So if I go back to my path variable, you can see that by doing a module load, my path variable was automatically appended with the right path, which allows the system now to find the BLAT program. So we obviously need some data files. Let's go over to our play area, netscratch your onion directory. So we're going to change directory to the netscratch area. Does anyone remember what we did, what command we had used last time to look at what the directory structure there looks like?
Like tree?
Yeah, tree. So remember, this is the dummy structure we had set up last time. My projects, we had created a few subfolders and then subfolders under there. So we're going to get some files and start learning how to move files between, let's say, your local desktop and the server. So for that, I want all of you to go back to our class resource site, and I have a few files here. We're going to learn first how to transfer files from the local desktop up to the server. Let's say I want to use the files called test.fa, which again, as you remember from last time, it's just a protein sequence in a FASTA format. And then db_prot, which is a slightly longer, again, multiple FASTA protein sequence file. So we're going to download these two files to the local desktop. And the way you're going to do that is by right-click on the file name and do save link as. Right-click, save link as. Let's see, it should allow us to put those on the desktop. It does. So I've grabbed one of the files and then I'm going to grab the second file in the same way. Right-click on the name, save link as. Again, go ahead and save it to the desktop. So I'm going to verify that both of those files are present on my desktop. We are using Secure Shell for interacting with the server, and there is a analogous protocol that goes with the Secure Shell program called Secure FTP, which allows us to transfer files between our local desktop and the server in a secure fashion. And we are going to use a graphical interface that is available in the Secure Shell program for doing that. If you hover over this yellow folder-like icon, you can see that this is going to open a new file transfer window. Now, just like we looked at last time, if I just click this icon, then you can see that it opens a secure file transfer window. So the left-hand pane here is your local desktop. The right-hand pane is the listing from the server. What it did is, by default, it puts you in your home directory. So the listing that the secure file transfer is showing you is from your home directory on the server. Now, since we are working in the netscratch onion directory, we're going to use the trick that we learned last time and not actually go up and down the directory structure here, even though you could. I'm going to close this window for a second. And since I am already in the netscratch Kelkar directory, I'll go up to the window drop-down box and say that let's go ahead and save a few keystrokes and open a file transfer window right in netscratch Kelkar, so we won't have to navigate around. So now you can see that it automatically puts us in the netscratch onion directory. So what I did was I went to window and I selected the second option, which opens a file transfer window in the directory that you may be in at any given time. It happens to be the netscratch Kelkar directory at the moment. Is everyone able to open that secure file transfer window in the netscratch directories? Okay. So at this point, transferring a file from your local desktop listing, which is on the left, to our server area is as simple as grabbing the file and dragging it to the right. Since I already have a file in my NetScratch area that has the same name, it's asking me if I want to override it. I'm just going to say yes. So and that's as simple as transferring a file from your local desktop to the server area that you're working in. You are obviously able to transfer entire folders this way too. You don't have to do single files. If you had a folder here with multiple files in it, you could just drag the entire folder and bring it over there. This is one area where you would want to keep in mind that you don't want to have spaces in the file names. It's easier not to have them. Underscores work much better. And this is the second file I need, the database file. So I'm again going to drag it over. I have a file there, so it's asking me if I want to overwrite it. I'm just going to say yes. So I'll go ahead and close the window for now. So this is one way of transferring files, by first bringing them down to your local desktop and then laterally transferring them. We'll learn a new way of doing it directly without having to bring them down in just a little bit. But first, let's go back and see if the new files show up. So you should have two files in there called .fa. Can somebody quickly tell me how do we find out what, for example, let's say the test.fa file contains? What kind of a file is it? What command did we learn last time? Okay, this is I think not one of the commands that you may remember. It's file will tell you what kind of file it is. So if you say file name, that tells you what sort of a file it is. We know that both of the files that we just transferred are just ASCII text files. Switching back to my slide here for a minute. We'll come back and look at these files, the other options here in just a second. So BLAT is a tool that you may have used through the UCSC Genome Browser page. It's available through that page, and that's where I tend to use it sometimes. This is a BLAST-like alignment tool that was designed by Jim Kent at UCSC. And the advantage of BLAT is, one thing is it's very fast. As compared to BLAST, you will find that BLAT returns search results very quickly. It's generally very useful to find sequence homologies when you have fairly homologous sequences that are at least 25 nucleotides or longer. And it does work with both DNA and protein. So the basic command line for running a BLAT search, so we've already loaded our module. So the way you can confirm that you have a particular module loaded is by using the command called module list. So module list tells us that we do have the BLAT module loaded and we have the BLAT program available. So as we remember from our first session, the Unix commands are always going to have the structure where you will have the name of the program, which happens to be BLAT in this case, and everything that follows are the command line options that that program takes. Some programs will require options, some programs will run without options. Basic system commands like ls that we've used or pwd do not need any options. They will still produce some output when run on their own. Now, in case of BLAT, you at least have to provide three minimum things. There has to be a file which is going to be used as a database. So this is the other advantage of BLAT. With BLAST, you remember that you have to create index files in a binary format, and then you are going to be searching against those index files. So there are index files for nucleotides, the non-redundant NRs, and you probably remember all of those from the BLAST searches that you may have run on the NCBI side. Now in this case, BLAT allows you to just take simple two text files and designate one of them as a database and use the other file to search against the first file that you designated as a database. So this is one advantage of using BLAT is you're able to use simple files without having to create indexes. There are obviously ways by which you can index a file for faster access. If you are going to run, for example, a BLAT search against the human genome, even though you could provide the human genome as a plain FASTA text file, there are ways by which a binary version of that file can be made available to BLAT. And then that we can probably talk about once we finish this basic use of BLAT. So first file that you provide on the command line always uses database. That's why the order that you specify for the files is important. The first file you specify will be used as the database. Second file will be used as the query file. And then the output will be written into the output file. Programs that are generally well written will actually try to help you. So if I just say BLAT and try to run it, it produces some helpful output that shows me that BLAT is a fast sequence search command line tool. It tells me what it is. It tells me how to use the program. So it's trying to help you be familiar with how you would run this program. And then as you go down, these are all the different options that the BLAT program can possibly take. You may end up using one or more or none. It doesn't really matter. It sort of is going to depend on what you're trying to do. But you can sort of read through these and they're fairly clearly explained as to what that does. It shows us that -t, you will, so wherever this word type says, you are going to obviously replace it with one of these words here, which means that -t equals DNA will tell the program that the file you're trying to use as a database is DNA of protein. Generally, the program is smart enough to figure that out, but you could specify that specifically. The one thing that we are going to actually use in our initial command line is this command called -out. So BLAT is able to produce output in various different formats that are pretty standard. So it can, by default, it's going to produce output in this PSL format, which is basically a tab separated file. And we'll look at these formats in just a second. But BLAT itself is able to produce output that just looks like a BLAST search. So if you prefer seeing the output of a BLAT search in a regular BLAST-like format, BLAT is able to do that. So without further ado, let's go to our shell and